With COVID-19 vaccines currently in the final phase of study, you've probably been wondering how the FDA will decide if a vaccine is safe and effective. Based on the status of the Phase 3 trials currently underway, it is unlikely that the results of these trials will be available before November. But it is likely that not just one but several of the competing COVID-19 vaccines will be shown to be safe and effective by the end of 2020. I am a scientist and infectious diseases specialist at the University of Virginia, where I care for patients with COVID-19 and conduct research on the pandemic. I am also a member of the World Health Organization expert group on COVID-19 vaccine prioritization. What is the status of COVID-19 vaccines in human clinical trials? Phase 3 studies are underway for the Moderna and BioNTech Pfizer vaccines and the Oxford AstraZeneca viral vector vaccine. Each of these vaccines uses the SARS-CoV-2 spike glycoprotein, which the virus uses to infect cells to trigger the immune system to generate protective antibodies and a cellular immune response to the virus. Protective antibodies act by preventing the spike glycoprotein from attaching the virus to human cells, thereby neutralizing the SARS-CoV-2 virus that causes COVID-19. In the case of Moderna's nucleic acid vaccine, the messenger RNA encoding the spike glycoprotein is encased in a fat droplet called a liposome to protect the mRNA from degradation and enable it to enter cells. Once these instructions are inside the cells, the mRNA is read by the human cell machinery and made into many spike proteins so that the immune system can respond and begin producing antibodies against this coronavirus. The found in chimpanzees shuttles the instructions for manufacturing the spike glycoprotein into cells. Studies in animal models of COVID-19 provide convincing evidence that vaccination with the spike glycoprotein will protect from COVID-19. Experiments have shown that when the immune system is shown the spike protein, which alone cannot trigger disease, the immune system will generate an antibody response that protects from infection with SARS-CoV-2. In studies in hamsters in adenovirus viral vector, the approach used by Oxford AstraZeneca, for example, was used to immunize with a spike glycoprotein. When the hamsters were infected with SARS-CoV-2, they were protected from pneumonia, weight loss and death. In non-human primates, DNA vaccines which deliver the gene for the spike glycoprotein reduced the amount of virus in the lungs. Animals that produced antibody that prevented virus attachment to human cells were most likely to be protected. The FDA has set the bar for the primary endpoint of a Phase 3 trial of 50% protection for approval of a COVID-19 vaccine. Protection is defined as protection from symptomatic COVID-19 infection, defined as laboratory-confirmed SARS-CoV-2 infection plus symptoms such as fever, chills, cough, shortness of breath, fatigue, muscle aches, loss of taste or smell, congestion or runny nose, diarrhea, nausea or vomiting. This means that an effective vaccine is considered one that will reduce the number of infections in vaccine recipients by half. This is the minimal protection that is anticipated to be clinically useful. That is, in part, because lower levels of efficacy could paradoxically increase COVID-19 infections if it leads vaccinated people to decrease mask wearing or social distancing because they think they are completely protected. Since a vaccine might be more effective at preventing severe COVID-19, the FDA instructs that protection from severe COVID-19 should be a secondary endpoint. Phase 1 and 2 studies by pharmaceutical companies Janssen and Merck also use viral vectors similar to the Oxford AstraZeneca vaccine, while vaccines by Novavax and GSK Sanofi use the actual spike protein itself.